Hello friends and welcome to today's lecture. I, Sunita Bundale, would be taking you today through termination and regulation of prokaryotic replication. After the extensive synthesis of DNA during elongation, which we saw in the previous video lecture, the process must be accurately and efficiently brought to a halt. This occurs when two replication forks progressing in opposite directions from the origin meet at a defined region on the chromosome, that is the termination site, thus concluding the replication cycle. Termination would mean dissociation of the entire replication machinery and also separation of the two completely formed daughter duplexes. Replication initiates at specific site called oriC. DNA replication occurs bidirectionally in E. coli. The two forks move in opposite directions. We've seen this in the previous video lecture. Where would the termination occur then? If there are specific sites for origin of replication, maybe there ought to be specific sites for termination as well. But where would those termination sites be in the genome? That is where the two forks would meet. These termination sites should be strategically located on the chromosome diagonally opposite to the origin of replication. Indeed, the termination sites called the ter sites are located diagonally opposite to the oriC. In E. coli, there are 6 well-characterized termination sites: ter A, ter B, ter C, ter D, ter E and ter F, which act as one-way barriers for the replication machinery. Each ter site is approximately 23 base pairs long, with the consensus sequence which you can see on your screen right now. There is a strictly conserved GC base pair at the 6th position, which is followed by a very highly conserved 13 base pair core region. The unique feature of the ter sites is that they are directional, that is, a replication fork can enter but cannot pass through from the non-permissive site. We saw that the initiation of replication along with the origin of replication, the initiator proteins, DnaA proteins, are also required to recognize the oriC. Likewise, it was thought that mere presence of ter sites would probably not be enough for termination. Probably some proteins are also involved. This led to the discovery of TUS proteins. TUS stands for termination utilization substances. The TUS gene lies 11 base pairs downstream of the ter B site. TUS protein is made up of 308 amino acids and has a mass of 36 Dalton units, around 36,000 Dalton units. TUS protein is a monomer, which is a DNA binding protein with an affinity for the ter sites. It binds tightly to each ter site, forming the TUS ter complex. TUS ter complex blocks the action of the major replicative DNA helicase. We will see this in the coming slides. Let us discuss the termination region of the E. coli genome. Two replicating forks with their accompanying replisomes are moving away from the oriC towards the terminator region on the opposite side of the circular E. coli genome. 3 termination sites operate for each fork. Ter E, ter D and ter A stop the counterclockwise fork, whereas ter F, ter B and ter C stop the clockwise fork. The TUS protein binds to the terminator sites and helps arrest the moving forks. Let us now talk about the mode of action of the TUS ter complex. The termination mechanism involves a protein called TUS that binds tightly to each TER site, forming the TUS-TER complex. The key role of this complex is to halt the progression of the DNA replication fork, particularly by interacting with DnaB helicase, the enzyme responsible for unwinding DNA ahead of the fork. The directional nature of this block is critical when the helicase approaches the TUS-TER complex from the permissive side. TUS is displaced and replication continues unhindered. However, if it approaches from the non-permissive side, TUS physically blocks the helicase, halting replication. This polar arrest is mediated by a molecular mousetrap mechanism. The unwinding DNA base at the junction inserts into a binding pocket in TUS, locking the complex and preventing helicase's progression. Let us discuss the replisome of E. coli and mechanism of replication of the replication fork arrest by the TUS-TER complex. The replisome moves along the DNA template and approaches the TUS, and the DnaB helicase assists the primase to lay down the last lagging strand primer. Next, DnaB helicase action is blocked by TUS and DnaB helicase dissociates from the template. After this, DNA polymerase III holoenzyme completes the leading strand synthesis up to the TUS-TER complex and synthesizes the last Okazaki fragment on the lagging strand, which will eventually be ligated by DNA ligase to the penultimate fragment following removal of its DNA primer by DNA polymerase I, which is not shown here. We have seen it in the previous lectures. The holoenzyme then dissociates, leaving a Y-forked structure that is single-stranded on the lagging strand near the TUS-TER complex. Thus, when the replication machinery dissociates, the two daughter duplexes are not completely formed and they are intertwined with each other. DNA replication often results in two completely or incompletely formed daughter duplexes intertwined on or interlinked with each other. These interlinked circular molecules are called catenates. They must become disentangled before cell division occurs, or they cannot separate to the two daughter cells. Separation of these catenates is called decatenation, and this is brought about by the action of topoisomerases. If the end result of the termination of replication is two incompletely formed daughter duplexes interlinked to each other, then they can undergo decatenation first. For this, a single-stranded nick is required, and this can be brought about by type 1 topoisomerases. The two incompletely formed daughter duplexes get separated. This is then followed by repair synthesis so as to yield two completely formed daughter duplexes. Alternatively, the two incompletely formed daughter duplexes can undergo repair synthesis first so as to yield two completely formed daughter duplexes interlinked to each other. This would be followed by decatenation, where a double-stranded nick would be required, and this can be brought about by type 2 topoisomerases. Having talked about termination, let us now talk a little bit about how E. coli undergoes replication before cell division and how its replication is regulated. This is an E. coli cell and this is its origin of replication. The replication has initiated and it takes 40 minutes to complete the replication of the genome of E. coli. It then takes 20 more minutes for cell division. Thus, it takes 60 to 70 minutes for one E. coli cell to become two. But I'm sure we have studied at some point of time or other that the generation time of E. coli or the doubling time of E. coli is 20 minutes. It means that in 20 minutes, the E. coli population doubles up. So how is this possible? If it takes 40 minutes just for the genome to replicate, how is it possible that the E. coli population can double up in 20 minutes? This is brought about by multiple initiation events in E. coli DNA replication. The second round of replication initiates before the first round is completed, so the daughter cells will have a DNA whose replication is already half done. As a result, the total time required for cell division will not be 60 minutes, but only 35 to 40 minutes. There can be multiple initiation events and multiforked DNA can be seen. The daughter cell with such multiforked DNA will just take 20 to 25 minutes to complete replication followed by cell division. But then there has to be some control on these reinitiation events. Otherwise, there would be E. coli cells with more than one genome in their cells. This control is brought about by methylation of adenines at the GATC sites. The GATC sites are palindromic sites which are present in the E. coli genome. The adenines in the GATC sites are methylated by an enzyme DnaA. Before replication, all adenines within GATC sites on both strands of the parental DNA are methylated by DnaA. Replication introduces normal non-methylated adenines into the daughter strands. Thus, after replication, each daughter duplex has hemimethylated GATC sites. These adenines would be methylated later by DAM methylase to form fully methylated GATC sites. Now, the two main sites of control of replication are oriC and DnaA gene. Within the oriC, there are 11 such palindromic GATC sites. Replication can initiate only at fully methylated origins. After replication, the oriC becomes hemimethylated origins which do not support initiation. These hemimethylated origins can initiate replication only after DAM methylase methylates the adenines. But how long does it take for DAM methylase to reach the GATC sites after replication? Well, there is a catch here. Almost everywhere else in the genome, DAM methylase takes just 1 minute to bring about methylation of the adenines after replication, except two regions. The first is oriC and the second is the gene for DnaA initiator proteins. At these sites, it takes 13 minutes for DAM methylase to methylate the adenines in the GATC sites. But why is this so? This is because the hemimethylated state of newly replicated oriC DNA is detected by a protein called seqA. seqA has an affinity for hemimethylated GATC sites and binds tightly to it. Binding of seqA has two consequences. First, it dramatically reduces the rate at which the bound GATC sites are methylated by preventing DAM methylase from reaching the sites. Second, when bound to these oriC proximal sites, seqA prevents DnaA from binding to oriC and initiating a new round of replication. Thus, the conversion of oriC proximal GATC sites from methylated to hemimethylated sites inhibits DnaA binding and therefore prevents rapid reinitiation of replication from the two newly synthesized daughter copies of oriC. But the binding of sec-A to hemimethylated oriC is temporal. The binding weakens after around an additional time of approximately 13 minutes and sec-A then dissociates from the GATC sites and leaves the sites open for DAM methylase to come and methylate the GATC sites. As a result, both strands become fully methylated, to which DnaA proteins can now bind and new round of initiation can be started. But have you realized that there is automatically an interval of 13 minutes in between two initiation events? Let us just quickly see again whatever we have studied just now. This is the fully methylated origin of replication. Replication occurs, and as a result of that, the region becomes hemimethylated. The hemimethylated GATC sites are attracted by sec-A. As soon as sec-A binds to the hemimethylated sites, it prevents the binding of DAM methylase for methylation of adenines. The binding of sec-A is temporal, and after a delay, the sec-A dissociates, leaving the site now open for DAM methylase to methylate the adenines and make the regions fully methylated. Just as oriC is one site of control of initiation of replication, the second target is the DnaA gene. The promoter of DnaA gene also has similar GATC sites which are methylated. In the hemimethylated state, the DnaA promoter is repressed and the level of DnaA protein goes down during this period. We know that the DnaA proteins are required to bind to the 9-mer sequences during initiation. Thus, the hemimethylated origins do not support the initiation at oriC and also the production of DnaA protein which is required for initiation, and it is not available in enough quantities for the reinitiation events to take place. With this, we come to an end of this lecture, where we have today talked about the termination of replication of prokaryotic DNA replication, and we have also talked about how the replication is regulated in prokaryotes. If you like the content of this video lecture, please subscribe to the channel. Thank you.